Okay, I'm going to show you some examples dealing with genetics, hopefully to explain a few things to clarify it. With genetics, we're going to be talking about the genes or the alleles. Remember, for every gene, basically, you have two copies because you got one from mom and one from dad. When we talk about specific genes, and on one chromosome you can literally have thousands of genes, we tend to give the genes a letter designation. And one of the classic things that when we talk about genetics that we'll often use is that if you're a tongue roller, if you can roll your tongue, that's genetic. Everything about you is determined genetically. So whether you can roll your tongue is genetics. So do you have a gene that allows you to roll your tongue? It's either yes or no. Rolling your tongue is what we call a dominant trait, and so just by tradition we would use a capital letter to designate that gene, and we usually use the first letter of the dominant thing. So in this case, tongue rolling is dominant, so we would use a capital T. Now, like I said, you have two copies. You got one from mom and one from dad. So if you can roll your tongue, you at least have one capital T, one dominant trait. Now, there is the possibility of having a recessive gene, which we will use as a lowercase t. That gene does not allow you to roll your tongue. We do not use a different letter because that's going to get really confusing real quick. So you use the same letter that you used up here to designate that you could roll your tongue, but you're going to use a lowercase letter for the recessive trait. The dominant one, as long as you have one copy of that gene for complete dominance, you will see that trait expressed. Now, if you have two copies of it, you'll still see it expressed. So since you get two genes, like I say, one from mom, one from dad, there's a couple of possibilities that an individual could be. They could have two of the dominant genes, big T, and that sounds strange initially, but that's very common when we talk about genetics, to have like big T, big L, little l, things like that. So you could have two of the big T's, two of the dominant genes. You could have two of the lowercase t's, or you could have one of each. It just depends on which genes you got from mom and which ones you got from dad. If the two letters are the same in terms of both capital or both lowercase, then that is referred to as homozygous. They're the same. Homo means the same. If they are different, one capital, one lowercase, then it is known as heterozygous. The capital letter designates that it's dominant, the lowercase that it's recessive. The only way with complete dominance, the only way that you will see this recessive trait, in this case, that you cannot roll your tongue, is if both of the genes that you have are the lowercase t's. Now, when we are talking about genetics, we also talk about genotype and phenotype. Genotype is what is on the genes. And genetics in a nutshell, what that means is what are the letters that you have. Phenotype is what do you physically see. Can you roll your tongue or not? Now, why do you care about this? Well, oftentimes what you're wanting to do is try to predict what the offspring potentially would be. So, just as an example, if I were to say that we had one parent who is heterozygous, so heterozygous means different, So it means there will be a big T and a little t, because we're looking at tongue rolling. And we are going to cross this with a parent who cannot roll their tongue. If they cannot roll their tongue, that means that they have 2 of the recessive traits. So how can we predict what the offspring would be? Can they roll their tongues or not? This is where we're going to set up the Punnett Square. So the Punnett Square is usually used to predict the probability in the offspring as to what their genotype and then what their phenotype would be. So we're looking at just one trait, so this is called a monohybrid. So what we're going to do is set up a Punnett Square. It will be a 2 by 2. We will take one of the parents. It doesn't matter who goes on the side and who goes across the top. One of the parents, we will take their genotype. We put one letter here and one letter here. This essentially is simulating what happens during meiosis in the production of the sperm or the egg, in that you end up with only half the number of the chromosomes. The other parent we're going to put down the side. Great, so you have one parent on the side, one parent on top. How do you figure out what the offspring are? Well, the offspring are going to be in the boxes. And to fill in the boxes, all you do is you bring the columns down and then bring the rows across. So in this example, what you would do if I were to ask, what is the genotype? You look at the letters. Notice in here you've got 2 of the 4 boxes, so 2 out of 4 is a big T, little t. And then 2 out of 4 are little t, little t. For the phenotype, as long as you have one big T for complete dominance, they can roll their tongue. So 2 out of 4 can roll their tongue, 2 out of 4 cannot roll their tongue. So this would be an example of complete dominance. Now we're going to move on to an example of what we call incomplete dominance. In this case, one trait is not completely dominant over the other. One of the classic examples of this is dealing with snapdragon flowers. If you have a big R, we're going to call that the red flower, the red snapdragon. And just a disclaimer here, this is true for snapdragons, but it's not true for all flowers. The flower color, yes, is determined genetically, but the way it works and which color is dominant, is there a dominant color, that is species dependent. Now, for sake of this, I'm going to show a different color here. Little r is going to be white. So, if you cross a red flower with a white flower, we're going to do a Punnett Square to see what those offspring are going to be. So if we put the red flower up here, so big R, little r, the other parent down the side. You still fill in the Punnett Square the same. Bring those columns down. Bring the rows across. Now, if we're going to ask you what the genotype is, you can see right here that 4 out of 4 are going to be the big R, little r. That is the genotype. Remember the letters. Phenotype. 4 out of 4 are going to be the same phenotype. But what is that phenotype? You have one red gene, one white gene. In incomplete dominance, one is not dominant over the other, and so it's like you get a mixing of it almost. They're both being expressed. And so what color would this be? In this case, the big R, little r, that is going to be pink. So that's an example of the incomplete dominance. I say the snapdragon flowers are probably the classic example that we use to show this. Now, have you lost that red and white gene? No, that's still there, because look what can happen. If we now take a pink flower and we're going to cross that with another pink flower, what do we end up with? We're going to cross these. Let's do a Punnett Square and figure that out. We're just looking at one trait, so it's monohybrid. We're going to put one parent up here, the other parent over here. We're going to bring, once again, the columns down. Usually you put the capital letter first. We're going to bring the rows across. Now, what do we have in terms of genotype? We have right here a big R, only 1 out of 4. We have 2 out of 4, a big R, little r. And then we have a little r, we have 1 out of 4. In terms of what the phenotype's going to be, the big R is going to be red. The big R, little r is pink. And the little r is white. So I said incomplete dominance. One is not dominant over the other. So you get this mixing, if you will. You get the phenotype is somewhere in between those two different traits. Another example that we have, erase this, is with multiple alleles where there's more than two varieties. In this case, you've had examples of the flower colors where there's been two possibilities. Red and white. Tongue rolling was either yes or no. For blood type, there's actually 3 different alleles, 3 different possibilities, but everyone only has 2 of those 3 possibilities. So you can have a big IA, you can have a big IB, or you could have a little lowercase I. Now, we also call this codominance because if you end up with the big IA and the big IB, you will see expression of both of those. They'll be equally expressed. But both of those will be dominant over the lowercase I. So, if a person is type A blood, so if we put the blood type here, if they are type A blood, what is their genotype? Remember the letters. They could be homozygous dominant. That means that both of the genes are identical. So it would be big IA, big IA. Or they could be heterozygous, which means different. So they have a big IA and they have a lowercase I. The lowercase I, remember, is the recessive one, but the big IA is dominant, so they would still have type A blood. If you are type B blood, you can be homozygous dominant, meaning the two genes are identical, or you could be heterozygous, meaning the two genes are different, but to have type B blood, you have to have the dominant and the recessive. Excuse me. If you are type AB blood, the only way this is going to work is if you have expression of these two genes. So you have to have a big IA to get the A component, and then you would have to have a big IB to get the B component. And then if you are type O, O is recessive, so the only way you're going to see that if it's lowercase I, lowercase I. Now, how would you do a Punnett Square to predict offspring with this? Well, let's see. You do it the same way. Let's say that we have an individual who is type A, and we're going to say she is heterozygous. So this would be her genotype. And then we are going to cross her with somebody who's type O. Now, even though there's two letters here, the big IA, this is considered one group, so you have to keep them together. So once again, we're going to do a Punnett Square because that's what we use to predict the offspring. And because there are so many different letters here, I'm going to use different colors. So if we take this parent right here, and we're going to put the big IA here and the lowercase I there, and then I'm going to use blue for over here. And put the other parent on the side here. Just like any Punnett Square, bring your columns down. And remember, this is a single unit, so bring them down together. Bring your rows across. Now, for the genotypes, once again, what do? You've got 4 squares here. 2 of them, so 2 out of 4 are a big IA, little I. That is their genotype. And then 2 out of 4 are little I, little I. Genotype is the letters. What is the phenotype? That would be the blood type that they are. So if they're a big IA, little I, they are type A blood. If they are little I, they are type O blood. There is one other example with genetics that I want to do, and that is what we call sex-linked or X-linked traits. Females have XX chromosomes, males are XY. The X chromosome is much larger than the Y chromosome, so there's a lot of space on there to have other genes that have absolutely nothing whatsoever to do with sex, but they're just placed on there. And some examples are things like male pattern baldness, color blindness, hemophilia, are examples that are on that X chromosome. Now, because males are XY, that means if there is some disorder that is caused by a recessive gene on that X chromosome, if they have it, they have it. They have that disorder. Now let's look at something that would not be, by most people, considered that devastating, but let's look at color blindness. Color blindness is on the X chromosome. So if you have normal vision, we'll do a capital C. You can see in color. If you have a lowercase c, which I'll try to distinguish this way, that means that's a color blindness gene. So for males being XY, this is what I'm saying. They either have it or they don't. If they have that color blindness gene, they're colorblind. Where did they get the X chromosome from? Well, if males are XY, they got the Y from their dad, they got the X from mom, which means mom is either a carrier or mom is colorblind. Females are XX. So here's what happens with females. There's actually 3 possibilities instead of 2. They could have 2 of the genes that allow them to see in color. They could be heterozygous, so they have one that allows them to see in color and one that for color blindness. In this case, this person is heterozygous. We also use the term that they are a carrier. Can she see color? Yes, she can. Does she have the color blindness gene? Yes, she does. Yes, she can pass that on to her offspring. The only way woman is colorblind is if both of the X chromosomes have the color blindness gene on them. Just FYI, if a woman is colorblind, remember, she got 1 gene from her mom, 1 from dad. Dad only has 1 X chromosome, which means if a woman is colorblind, you know that her biological father is also colorblind. And then mom would either be colorblind or could be a carrier. So how would we set this up with a Punnett Square? Let's say that we have a woman who is heterozygous. So she has 1 normal gene for color and 1 color blindness gene. And we are going to cross her with a male who, let's say, is colorblind. Let's predict what's going to happen with their offspring. Do a Punnett Square. Once again, you put 1 parent up here. The C is going to be attached onto that X chromosome, so you have to keep it as a group. So once again, let's bring columns down. And we'll bring those rows across. That's supposed to be a C. So now, if we look at this to predict what the offspring are, XX are females. So if you want to you can word it a couple different ways. You could say that this individual is a female, has 2 X chromosomes, has 2 of the lowercase c's, so this individual is going to be colorblind. This individual, it's a male, XY. The X chromosome has a lowercase c on it and will be colorblind. So you could say out of the possibility of 4 children, the 4 possibilities, 2 out of 4 would be colorblind. 2 out of the 4 can see color. This would be the phenotype. You could also distinguish if you wanted to and just say 2 or 50%. If you look at the 2 females up here, 1 out of 2 or 50% would be colorblind. Same thing for the males. Of the 2 potential males, 1 could be colorblind or 50%. This male has normal color vision. This female has normal color vision, but would be a carrier.